Okay, so this is now mini lecture 2. It's shorter than the first one, so let's get going. So it's important to distinguish between the cyclic GMP dependent effects in vascular smooth muscle, which are outside the muscle sarcolemma, versus muscle intrinsic function of nitrosylation. So sarcolemmal nNOS, as I mentioned, produces NO right at the sarcolemma, can diffuse into the interstitium, and hope I've convinced you can recreate normal functional sympatholysis and improve post-exercise hyperemia, which is blunted in the patients with Duchenne Becker. But nitric oxide can also simply put a NO moiety on other proteins, in this case HDAC, histone deacetylase, in skeletal muscle, and that's key for blocking inflammation. Let me explain that in more detail. So the nitrosylation has nothing to do with cyclic GMP. This is just a direct chemical reaction. So this paper from the group in Italy that was very nice, and it suggested that normally one of the things that sarcolemmal nNOS does is that by nitrosylating one specific HDAC, that it removes, HDAC is normally suppresses miRNA promoters that are very important to block inflammation. So you have to have sarcolemmal nNOS to nitrosylate HDAC to take it off of the promoter and let the promoter do its thing. The NO is produced at the sarcolemma, it diffuses into the cytosol, binds to nitrosylates HDAC, so that's a block against too much inflammation. In the dystrophic muscle, with the absent sarcolemmal nNOS, HDAC is not nitrosylated. It blocks the production of these good miRNAs and leads to has a permissive role to allow too much oxidative stress and fibrosis in the muscle. So that's the theory based on mainly in vitro work and in some in vivo mouse work. Does it have anything to do with human beings? So how about a chronic NO donor for Duchenne or Becker? Well, will this correct the abnormal blood flow regulation? I'm going to show you the answer is yes. Will it restore dystrophin? Absolutely not. But there is some precedent literature that this might, based on mouse studies, it might upregulate utrophin, the dystrophin homolog. It may activate satellite cells, which is the muscle's resident population of stem cells, and therefore increase muscle mass, and reduce inflammation and fibrosis, like I showed on the previous cartoon. Okay, so this is work from my friend Raju's lab. I can't ever pronounce his last name, so he calls himself Raju. This is in Washington, D.C., and they do, this group does fantastic preclinical trials in the mouse. One of the reasons that he believes that it's been so hard to translate work from mice to men and boys with Duchenne is that most basic scientists, no offense to anyone in the audience or their faculty members, are based on very small n's and don't use the same rigors that we have to use in clinical trials with blinding and adequate sample size in large numbers over a long enough time of treatment. So this is 9 months of treatment in an MDX mouse. They're treated, it's either wild type treated with vehicle, or the MDX mouse treated with vehicle or an NO donating non-steroidal anti-inflammatory drug. So what this company did, this naproxenol, they took naproxen, which is a leave, the same drug I'm taking after my oral surgery yesterday to block inflammation. And so a leave, you can buy over the counter. What they did was they with some proprietary chemical rearrangement, they added an NO donor moiety onto the Aleve. The original reason to do this work was to block some of the cardiovascular toxicity and GI toxicity, ulcerogenic toxicity of these non-steroidal anti-inflammatory drugs. But it also could be repurposed for muscular dystrophy, and this this compound has not yet gotten FDA approval for any indication. But here in this preclinical mouse study, after 9 months, what they're doing here is measuring cathepsin activity. It's in vivo marker for inflammation. So the MDX mouse on vehicle has a lot of inflammatory stuff in this light blue-green color. And with this NO donor, it's either the NO that's being helpful, or it's the non-steroidal anti-inflammatory activity, the parent molecule, is blocking inflammation, as well as prednisolone, which is a standard glucocorticoid. And that's the point of this optical imaging slide. And what really got my attention was that after 9 months, they have 3 different doses of this NO-donating NSAID, or NO-donating Aleve, and they showed that muscle mass increases in the MDX mouse, but actually doesn't increase and tends to decrease with prednisolone. So this is fantastic. The only way you're going to increase, and grip strength increased. You know, the forelimb ability to grip strength, like the equivalent of our maximal voluntary contraction, actually improved with this compound. The only way to do that is to bulk the muscle, and you can see that muscle mass increased. They also improved cardiac function. I didn't show that on the next slide, on that slide. But the $64 million question is, can we translate this to patients? So to begin with, we wanted, Gail used her model of functional sympatholysis in the anesthetized MDX mouse. So now these are 2 different doses of norepinephrine at rest and with muscle contraction. You can see the impaired sympatholysis at both levels of norepi in the untreated MDX mouse. This is 1 to 3 months of therapy with 2 different doses of the prototype NO-donating nonsteroidal anti-inflammatory drug. And you can see that with the low dose, there's beginning to be some restoration of sympatholysis. And with the high dose, there's obvious rescue of sympatholysis in the MDX mouse. So it's a dose-dependent correction after weeks of treatment, and very exciting. So then, can we translate this to human patients? How do we think this works? Well, normally NOS, nitric oxide synthase, requires oxygen to generate nitric oxide. So this is the basic nitric oxide biology. So L-arginine is a substrate for NOS, requires oxygen to produce nitric oxide, and citrulline, the latter thought to be inert. Then what's key is that nitrate, NO2, thought in the past to be just a breakdown product of nitric oxide, can actually serve as an alternate donor of NO under certain circumstances. Those circumstances are, it requires deoxyhemoglobin to generate nitric oxide. So that the nitrite plus deoxyhemoglobin plus hydrogen ion leads to the generation of nitric oxide and these other products. And deoxygenation is exactly what happens in normal skeletal muscle during exercise. And if you remember what I showed you from the near-infrared spectroscopy, that happens excessively in the patients with Becker and Duchenne. And what got my attention was there's this great paper in the journal Blood that said that deoxymyoglobin works 36 times faster and more efficiently as a nitrite reductase as does deoxyhemoglobin. Well, myoglobin is only present in muscle, in skeletal muscle and cardiac muscle. So that I think that this is a perfect way for an NO donor or nitrite to function as an NO donor. So here's the paper this was several years worth of work. Mike Nelson is again the first author, and it shows that sodium nitrate, oral simple NO donor, not combined with an NSAID, alleviates functional muscle ischemia in patients with Becker. So here, first of all, before we get into the therapy, we sent the paper off to Journal of Physiology London, and the review came back and said, this is all very nice, but you need to bring back as many patients as you can, biopsy them, and prove to us that not just in one pair of patients as in your Science Translational Medicine paper, but what's the real relationship between presence or absence of nNOS at the sarcolemma and blood flow regulation. So here are patients. I think we have 13 patients on this slide. We ordered them in different number than they came through our lab. The first 7 had no nNOS staining at the sarcolemma, and the protein content was way down. This is percent of normal, 100% being normal, and you can see how low the protein content was. Most of these had deletions of exons 45 through 47, or 45 through 48. But there were 2 with deletion of exons 3 through 7, and in this case there was no sarcolemmal nNOS. And sympatholysis was greatly impaired. Normal sympatholysis is shown on the right in the healthy controls, and to show the range of it, the normal subjects, the percent attenuation is anywhere from 33% to complete attenuation, the average being around 50 to 60%. There's no overlap in these first 7 patients between the exercise-induced attenuation of reflex vasoconstriction in the Becker patients that can't bind nNOS to the healthy controls. So this is the biggest cohort we have. And then the other patients, after patients 8 through 13, have other deletions besides the ones we could predict. And there, of 6 patients, 4 of 6 had preserved functional sympatholysis, and 2 didn't. So again, sympatholysis follows sarcolemmal nNOS. So what about these exceptions? 9 and 13 are first cousins. These are men in their 20s that have a weird splice donor mutation of exon 46. It's not a whole deletion. One patient had plenty of nNOS at the sarc. Let me see here. Had plenty of. They both had sarcolemmal nNOS, but they were discordant for sympatholysis, preserved in one, but greatly decreased in the other. The cousin that had the preserved sympatholysis had a normal protein content, whereas the one that didn't had a very low protein content. So, that sort of goes. So for a human biological correlation between, if you will, structure and function, this is about as good as it gets. So it was very reassuring to us and to the reviewers of the paper. So that's before we treated anybody. So then in the treatment, we only took the patients that had absent sympatholysis at baseline to go to treatment. So because we didn't have access to the NO-donating NSAID, anyway, we wanted to get away from that NSAID moiety because that could confuse the picture. This is a simple NO donor. This is beetroot juice. We get nitrates in our diet. They've gotten a bad rep for possibly causing cancer, and that essentially has been debunked, or the data don't show it. You think about that, and you go to a kosher deli, and you get the meats have a lot of nitrates in them. This is from beetroot juice. A lot of the beets have a huge source of nitrate, and it's been concentrated here by a professor in Oxford in the UK. And so the nitrate, the thing is, when you swallow it, you swallow this juice. By the way, you could buy this at Whole Foods. It comes in 450 mLs, and it'll take up. It just tastes terrible. I couldn't get one glass down. This stuff tastes better. It's real sweet, and it's only about 75 mL, so you can just drink it. And it's called the sports shot because they think you'd use it to improve athletic performance, the way blood doping might. Anyway, when you swallow this stuff, the only problem is you need commensal bacteria in the mouth to convert nitrate to nitrite, because that's what's taken up into the bloodstream. So if you brush your teeth with or take antibacterial mouthwash, this stuff won't work. And if you spit all your saliva out, it won't work. So here, so that's the only thing about it. But so we asked the subjects in the lab, don't brush your teeth, don't use Listerine in the morning of the study. Okay. So this is there are three, it's actually yellow beets, this is red, it's close enough. These are three big, humongous beets that are mashed up and condensed into this, this concentrated juice. And this is the beetroot sport shot. And what's cool about this is that the professor at Oxford that made this has a placebo where it tastes exactly the same but takes all the nitrates out of it. So it's really good for doing clinical research. So will sodium nitrate beetroot juice restore some patholysis in patients with BMD? And this is one patient which you can see it did. So it's really interesting. It takes 3 hours to get peak absorption of tadalafil. It takes 3 hours to get peak absorption of sodium nitrate, and you don't need a prescription for it, and it's a heck of a lot cheaper. So here's, and just as an aside, you know, if you ask the patient, the parents of the boys with Duchenne, you know, are you taking nutraceuticals, herbal supplements? And there's so much use of herbal supplements among these desperate families without any evidence of whether they're good, bad, or indifferent. Here's a nutraceutical that I think could actually have some therapeutic benefit. So he has impaired some patholysis in a young man with Becker muscular dystrophy in his early 30s. We give him this one sport shot of sodium nitrate, and now you can see that this patholysis is restored. Looks just like a tadalafil study. Here's the summary data for, I believe, a dozen patients. And so here's impaired some patholysis at baseline, and after sodium nitrate, there's about a 50% attenuation, which looks like a normal response. So just like tadalafil. Now, here's the patient-specific data, and I'm going to ignore patient 5 over in right field. We'll come back to that. So the other patients, you can see that at baseline, that all but one, patient 10, had impaired some patholysis. Almost every time we do this, to begin with, we're going to find 1 or 2 patients that have preserved sympath, and the rest it's impaired. Back to sodium nitrate, it worked in all these patients to restore some patholysis. And what you see, rather than at the bottom, we have the change in the blood level of nitrite. And these bloods are sent to Jason Allen's lab at Duke. He's now in Melbourne. He has the best lab in the planet for measuring plasma nitrites. He can separate the nitrate from the nitrite, and that's key because the nitrite is the active ingredient. So the increases over baseline, the lowest was 55 and the highest was 330. Okay, patient 5. Patient 5, we studied him twice. The first time we studied him in visit 1, he had impaired some patholysis, and he was resistant to sodium nitrate. We get the nitrite level back, and it's even lower. It's 42. And we asked him, well, what happened? And he fessed up. This is a Texan. He's from Dallas, where I'd spent 23 years. And leave it to the Texans. He was a bad boy and brushed his teeth and used Listerine after we told him not to. So he didn't do what we asked. After 6 months of asking the IRB, we finally got IRB approval to bring him back. Why did it take 6 months? Because we asking him to do extra work compared to the initial informed consent. So he comes back on visit 2. Baseline, again, he has impaired some patholysis, and now he behaved himself, didn't brush his teeth or use Listerine in the morning of the study. We bumped up his plasma nitrite level to twice that from the first study, and ta-da, now we have rescued some patholysis. So he had randomized himself to the control arm of the study, and it's sort of a cute case report, if you will. Okay, then, so Ryan Rosenberry learned from Shu how to do these Doppler studies. Ryan is a co-founder of the Bruin Allies for Duchenne, And when I gave this live lecture for Rochelle's class, Ryan came up to, with one of the students came up after the class, and he worked for us as an intern for a year, did such a great job that after he graduated, we hired him as a full-time research coordinator, and he's been in my lab now. This is his second year. He's done just unbelievably fantastic job, as he prepares to go off to med school. Anyway, so these are the studies. This is the Doppler blood flow from rest to immediate post-hand grip exercise. On placebo, the patients, this subset of patients, this is placebo, double-blind randomized crossover trial. So I think 6 patients here came back and took part in this placebo-controlled study. And so here, there is an increase in blood flow in all 6 patients. When we give the sodium nitrate, there's no effect on resting blood flow, but the increment post-exercise improves in all the patients. And that's really important because it shows that what sarcomeral nNOS does is it's inactive at rest when there's no contractile signal. And remember that sarcomeral nNOS is a calcium-dependent enzyme, so as with increasing muscle exercise, muscle work, there's more NO produced. So it's very cool that the beetroot juice doesn't increase anything to resting blood flow, but increases post-exercise flow. What about healthy controls? This is the increase in blood flow from rest to exercise in healthy controls. It's much more than the patients. Sodium nitrate doesn't do anything for their blood flow. In fact, there's a tendency that it blunts it. And so it's really interesting because the company that sells, this company and the other companies that advertise beetroot juice on the internet are selling it that it's going to enhance muscle performance in normal, in fact, in trained athletes. Well, this data, at least when it comes to blood flow, that ain't the case. So what it means is that once sarcomeral nNOS is plentiful, the beetroot juice doesn't do anything to further improve blood flow in these patients. So here's, so we took our near-infrared spectroscopy, and instead of looking at the oxygenated signal, we look at the deoxy signal. So this near-infrared can't separate hemoglobin from myoglobin. The spectra are too close. But this is the deoxy myoglobin signal, and this is the change in it from placebo to sodium nitrate. There's really no significant effect. Slight increase maybe with sodium nitrate, but it goes up, which is what we expect because this is mild exercise at 20%. The plasma nitrite goes up a lot when we give, of course, the beetroot juice with sodium nitrate. So the 2 conditions required to have the nitrite, the sodium nitrite, become an alternative NO donor is that there's increased concentration of deoxyhemoglobin myoglobin, the reductase, and increased concentration of the substrate, which in this case is sodium nitrate, or sodium nitrite. So proof of concept, so here's what we think is going on. When we give the sodium nitrate, it's converted to nitrite. And so we think that it gives off its NO right at the sarcolemma. The deoxyhemoglobin is on the, if you will, the outside the muscle cell on the vascular muscle side of the sarcolemma in our way of thinking of things. And the deoxymyoglobin is right at the, inside the muscle, so that the NO released is right at the sarcolemma and sort of recapitulates the action of sarcomeral nNOS. So this is so simple, it's almost too simple to be true. But there's no engineering here, there's no transgenics. This is just a simple, silly juice that may recapitulate a lot of the work, the activity of sarcomeral nNOS. So that concludes the second mini lecture, and stay tuned. In part 3, we'll then take this work from proof of concept to clinical trials. Thanks for your attention.